Morning everybody, we are from Group 3 and this is our assignment video for the subject Veterinary Public Health, and our topic for this video is on Cryptosporidium. The linear classification for Cryptosporidium are as follows. The kingdom is Animalia, phylum is Apicomplexa, the class is Sporozoocida, the order is Eucocidiorida, family is Cryptosporididae, genus is Cryptosporidium, and the species is Cryptosporidium species. So here comes the introduction on Cryptosporidium. Cryptosporidium is a hardy protozoa which resides in small intestine. They are resistant to many disinfectants too. Apart from that, Cryptosporidium are responsible for many clinical disease in domestic animals and also humans. Cryptosporidium is associated with outbreaks of diarrhea in calves, lamb, kids, piglets, foals, young dogs, and also cats. The species of Cryptosporidium that affect poultry are Cryptosporidium bailei and also Cryptosporidium meleagridis. Here are some important notes on Cryptosporidium parvum. Cryptosporidium parvum will cause neonatal diarrhea in calves and lambs, and also Cryptosporidium hominis will cause respiratory disease in poultry and game birds. Cryptosporidium are transmitted by two ways. The first one is by direct contact with infected animal, while the second one is indirect transmission, which involves contact with Cryptosporidium fecally contaminated material through environmental contamination and also inhalation of the oocysts. And also note that Cryptosporidium does not spread through contact with blood. The protozoa Cryptosporidium does not enter the cells of the host, and it also lacks of host specificity, which means it can infect different types of species of animals. Animals can also be infected by Cryptosporidium by ingestion of the oocysts through contaminated food or drink. Cryptosporidium can also cause extraintestinal infection like respiratory tract infection, biliary tract infection, pancreas infection. Cryptosporidium tend to infect the brush border of epithelial cells, mainly in gastrointestinal tract. They can also cause death in immunocompromised individuals, and they can also cause life-threatening diarrhea. In this slide, it shows that there are different species of Cryptosporidium with different hosts and also with different predilation sites. Here comes the life cycle of Cryptosporidium. Cryptosporidium parvum and also Cryptosporidium hominis are spread mostly by contact with contaminated water. Food sources such as chicken salad may occasionally serve as transmission vehicles. Zoonotic and anthroponotic transmission of Cryptosporidium parvum, as well as the anthroponotic transmission of Cryptosporidium homini, occur through contact with infected animals or contact with water contaminated with infected animal species. Assistation of Cryptosporidium occurs after ingesting by an appropriate host. The sporozoids of Cryptosporidium are discharged and parasitize epithelial cells in the gastrointestinal system, as well as other tissues such as the respiratory tract. In these cells, the parasites, which is Cryptosporidium, they multiply asexually by schizogony or merogony, and then sexually by gametogony, producing microgametes, which is male, and also macrogametes, which is female. All fertilization of the macrogametes by the microgametes oocysts form and sporulate in the infected host. There are two types of oocysts produced. The first one is thin wall, which is commonly excreted from the host. The second one is the thin wall, which is primarily involved in auto-infection. The oocysts of Cryptosporidium will become infective upon excretion, and it allows for the direct and immediate fecal-oral transmission. This picture shows the life cycle of Cryptosporidium that we retrieved from CDC. As we can see, firstly, the cattle are major hosts for Cryptosporidium parvum. The thick wall oocysts, which is the sporulated form, will exit the host. Then it will contaminate the water and food. After that, the thick wall oocysts will then be ingested by other hosts. The pathogenesis of Cryptosporidium starts with a host infected with Cryptosporidium oocysts via fecal-oral route through direct contact with infected animal feces, or indirect contact with infected animal feces via contaminated water. Then it follows by ingestion. The oocysts will undergo sporozoid excitation. Then the sporozoids will infect and parasitize the gastrointestinal epithelial cells of the host. After that, it will cause the increase of intestinal barrier, intercellular permeability, and cause submucosal layer inflammation. If the host is immunosuppressed, Cryptosporidium will produce a severe life-threatening diarrhea and also large volume of stool. It will also cause severe dehydration and shock, and eventually death.
The clinical signs that we can see in animals that have been infected with Cryptosporidium are water diarrhea, dehydration, lack of appetite, weight loss, stomach cramps or pain, fever, nausea, and vomiting. There are a few ways that we can perform diagnosis on the animals that have been affected. with Cryptosporidium. Diagnosis method that we can use are wax mount, trichome test, and modified acid-fast staining. In this slide, it shows Cryptosporidium species oocytes in wax mount. In wax mount method, oocytes of Cryptosporidium can be observed. The pink arrow on the picture on the left side shows the oocytes of Cryptosporidium in wax mount, and the brown arrow is the budding yeast located in the same field as the oocytes. On the right side is the image of the Cryptosporidium parvum oocytes in wax mount. They can be seen under differential interference contrast microscopy. In this slide, it shows the Cryptosporidium species oocytes in trichome. Here, the image on the left side shows that the Cryptosporidium oocyte is represented by a red arrow. The blue arrows represent yeast. The image on the right side shows the trichome staining is inadequate for a definite diagnosis because oocytes will appear unstained. Cryptosporidium oocytes are represented by red arrows. Blue arrow represents yeast. In this slide, it shows the Cryptosporidium species oocytes stained with modified acid-fast. The image on the left side shows, against a blue-green background, the oocytes stand out in a bright red stain. Sporozoites are visible inside the two oocytes to the right in this image. The image in the middle displays the Cryptosporidium parvum oocyte in wax mount. They can be seen under differential interference contrast microscopy. The image on the right side shows Cryptosporidium species oocytes stained with modified acid-fast. Additional notes: Cryptosporidium species oocytes are rounded and measure 4.2 to 5.4 micrometer in diameter. Sporozoites are sometimes visible inside the oocytes, indicating that sporulation has occurred. Staining of Cryptosporidium species oocyte can be variable. In particular, infections that are resolving can be accompanied by increasing numbers of non-acid-fast oocytes. Goes treatment and control. There is no known treatment that available for symptomatic treatment. Antidiarrheals and fluid replacement therapy can be given. Haloperidol is available for the prevention of Cryptosporidium in cats. Phyllocin, azithromycin, or nitazoxanide were applied in cats. Infection is difficult to control since oocytes are hardly resistant to most disinfectants, except formicillin and ammonia. Prevention: practice good hygiene and purifying drinking water, and help prevent the spreading of Cryptosporidium. Avoid animals' fecal exposure. It is also a way to prevent the Cryptosporidium infection. On poultry farms, litter should always be kept dry, and special attention needs to be given to litter near water fronts or feeding throughs. Feeding and watering utensils should be of such a type and height that droppings cannot contaminate them. Risk factors: the ones that are in risk of infections are children, animal handlers, those who drink untreated and unfiltered water, swimmers that swallow water in pools, lakes and rivers, the travelers that travel to developing countries, people who drink water from shallow and unprotected wells, and also parents of affected children. Complications that can occur due to the infections are malnutrition due to poor absorption of nutrition, so known as malabsorption. Next is severe dehydration, significant weight loss, inflammation of bile duct, gallbladder, liver and pancreas, and also weakened immune system tend to develop dangerous complications that can be life-threatening. Thank you. The video are prepared by Group 3 PVN4.